Hi everyone. So actually, I'll talk about a very related research topic. And thanks to the previous speaker, now I do not need to do the general introduction slides. So, but we all attack this problem from a different angle. So, well, this is the brief or introduction of why we're interested in homo-oligomer. As the previous speaker has mentioned, homo-oligomers are involved in many important biological functions like allosteric, pH-dependent binding, catalytic site interface, competition, shape, like forming the barrel, ring, pore, cage in the cell, scaffolding. It has a variety of important functions in cell. That's why we're very interested in it. However, we know that, as the previous speaker has mentioned, when you try to predict a giant homologomer structure with AlphaFold, you will have to first know how many copies you have, but then also when you have so many copies, AlphaFold scales, the computation time scales very badly with the number of copies. And then we thought, okay, why not take a step back and think about how our co-evolution information encoded in the sequence? And we know that something that's so in the very beginning of structure prediction field, people first look into residues are co-evolving when they do multiple sequence alignment. And then later, people look into like things like protein language model, that they just mask out the residue and then ask the model to recover the residue. And then for things like AlphaFold, you have to input MSA, which encodes the co-evolution information. One thing we ask is that, okay, so for homo-oligomer, actually, even if you just have the single sequence instead of giving multiple sequence for homo-oligomer, it should have encoded the co-evolution information in that single sequence, because like if you like in this case, say this is the structured homo-oligomer, you will probably have like intro contact encoded in the co-evolution information in the sequence. But then you will probably also have the inter information also encoded in the sequence. Like so then we thought, okay, then maybe even if you just give the protein language model the one single copy of the sequence, it probably will also give you contact for the contact between each of the chains. So we look into this very simple protein that's actually used the example on the AlphaFold collab notebook. And what we realize that the structure, the contact information output by ESM2, one of the most well-known protein language model, is actually a mixture of the inter and intra contact. Like you can see here, this is an intra contact of each within each monomer copy. And this one is basically intercontact between each of the copies. And here, if you look at the ESM2 contact map, you see what it's predicting is basically a mixture of the intra-contact and intercontact shown in red. And this is ESM2 model without any retraining. So this is a very interesting observation, because for a very long time, people have been using the predicted contact map of ESM2 as the contact map of the monomer. And when they train the contact head of ESM2, they train it with monomer contact maps. And actually, that's not true. A lot of the contacts you might label as false contacts are actually homo-oligomeric contacts. And so then after this very encouraging example, we started looking into analyzing basically all the homo-oligomers in the PB database. And we look at how can we improve the prediction of this intercontacts? And we realize that as people have observed for intra-contact prediction, the intercontact prediction with protein language models also scales as the model scales up. Like when you have the 8 million parameter model, it's barely predicting any good intercontact. But with the ESM2 3 billion parameter model, you have like quite good intercontact prediction. And then the ESMC, the newly released one with 6 billion parameter, has significant improvement over the 3 billion parameter one. And here, if you look at the contact map, this is an experimental contact map for this structure. In the ESM2 3B model, the previous version, you have no intercontact predicted. And in ESMC 6B million model, you have this contacts predicted. And I also want to highlight that even though you can see the intercontact prediction accuracy seems to be much worse than intracontact one, actually the good the good news is that we don't need that many contacts to be recovered. Like here, maybe you have around 30 contacts, and then here you recover like only maybe around 10 of them. You might say, okay, this recovery rate is very bad, but then actually, since you already have the monomer structure, you only need probably like 4 contacts to figure out how to dock the monomer to form the homo-oligomer structure. And another thing we thought is that, oh, maybe we can fine-tune the model better, because as I pointed out previously, when the ESM2 people train the models, for training the contact head, they only use the monomer contact map to train the contact head, which is wrong. And we noticed that if we add LoRa and to the model, and if we add, if we unfreeze the contact head to fine-tune the model, we can improve the performance of the model. And if we train it with only the monomer contact map, then performance is much worse than if we train with overlay of the inter and intra contact map. And here we see that we show example of this homo-oligomer complex. If you, with the original ESM2 3B model, these are the contacts they predicted, and with our fine-tuned model, we have recovered much more contacts, as shown in the red here. So these are great. Like, okay, it looks like we can improve the looks like, okay, first protein language models have intrinsically learned homo-oligomeric contacts. And secondly, scaling up the model really helps with performance, and fine-tuning the model really helps the performance. But then we're not very satisfied with the results, because we're like, why are all those intercontacts missing in these examples? When, like, we were very excited at first for the first example we saw that like in this example, okay, all the intercontacts were basically there. And we're like, okay, why when we look into more homo-oligomers, frequently we have missing intercontacts? So we went more into that direction to understand why the intersignals are missing. So one thing we thought is that actually, so for some homo-oligomers, the interface is like really tiny. So in that case, if you change the residue a tiny bit, maybe it just directly fails. So you have no room for convolution to happen, which requires changing of residue pairs. And so then we analyze, okay, whether some of the residue pairs are interface actually too conserved. And we did see that some of the residues pairs that we have missing signals in the protein language models, actually at least one of the residues in the residue pairs are too conserved. So that residue is not changing at all. So then you would lose co-evolution signal. And also we checked with a very simple method, which is just calculating the inverse covariance of MSA. This removes the indirect contact and give you the direct co-evolution, direct contact between residues. And we noticed that basically a lot of intercontact were missing, the signals are also missing in the MSA. So we know it's not a protein language model problem, rather it's like it's just general missing in the multiple sequence alignment. Secondly, we thought, okay, we talk about, okay, maybe some of the homo-oligomer interfaces are very tiny, that the residues are not changing at all, that's very conserved. Another question we thought is that people have been telling us about there's transit interaction, obligatory interaction. So we thought, okay, maybe some transit interactions are harder for protein language models to capture the co-evolution information. So one simple parameter we look into is a PISA dissociation energy score from the PB database. So in the PDB for homo-oligomer structure assignment, you usually have 2 tags. One is whether it's assigned by the author. The second one is whether it's assigned by the PISA dissociation energy. One has something that's above 0 is considered a homo-oligomer. And it's also giving some dissociation energy of like between -10 to 0 for some homo-oligomers, because it's like not very sure whether it is a monomer homo-oligomer. And what we realize that so when you have a PISA dissociation energy below 10, frequently you have no intercontact predicted by protein language model. And if you have anything above 10, then it doesn't really matter. The performance is not very different when you have a, say, dissociation energy of like 15, or when you have like 70, 80. So we do see that, okay, interprediction performance is slightly worse for proteins with PISA dissociation energy below 10. And second thing we noticed, and as pointed out by previous speakers, that actually in the PDB database, sometimes you can have proteins that belong to the same family. Like here, all these 3 proteins belong to the response regulator family. The monomer structures are almost the same, but then they actually have 3 completely different homo-oligomeric interface, shown in the red, blue, and yellow dots here. And we can see that actually protein language models, they're predicting a mixture of the 3 different interfaces when you give the sequence of each of the PDB. So we think this might be one of the reasons that the protein language models are missing some of the intercontacts, because the intersignals are diluted in the things like multiple sequence alignment. And then we check, okay, whether that is like a single case or it's actually a very common case. So here I look into the PDB database, which provide the sequence identity cluster. And what I have realized that, actually there are 2 very interesting things here. First thing is, as the audience has just pointed out, that even when you have 100% sequence identity, sometimes you can have different homo-oligomers. And I checked the PDB database. It can be caused by many different factors. And one factor is, as the audience pointed out, when you have different conditions. So, and this is one first interesting observation. The second interesting observation is that if you look at PDB sequence identity cluster of 30% sequence identity, which is what people consider to be sequences that are homologous to each other, sequences that belong to the same protein family. And you see that more than 60% of the cases you actually have more than one more than one different symmetry of these proteins. So you can see that this is why if you use methods, whether it's based on MSA or method based on protein language model, they're likely gonna get wrong answer, and you're likely gonna lose intermonomers signals, because in your MSA you're gonna have proteins with very similar sequence identity but actually have different symmetry. And another thing that I want to point out is that if you look into the PDB database, a lot of people when they train the model, they use PDB database as if that's like the ground truth. However, what we realize is that we find out sometimes even when the other assigns something to be a homo-oligomer, if you look in the paper, like if you check the protein purification curve from like HPLC, you will realize that it's actually a monomer. So after we see that case, we decide to do a more thorough analysis of the paper. So here what I did is I look into cases where basically I look into a group proteins where there's no intercontact predicted by protein language model, and I also look into group proteins where the intercontact prediction are pretty good. And here I parsed around 200 papers, and I noticed that for around 90 of them, or around 120 of them, 90 to 120 of them, I managed to download the papers, and then I use GPT API to parse the paper to extract relevant information. So what we have noticed is that for the cases where there's no intercontact predicted from protein language model, 1/3 of the cases actually in the paper is mentioned to be a monomer, even though in PDB database it's assigned to be a homo-oligomer. What does this mean? Like here I've shown 2 examples. The first one is with PDB ID 1GQP. It's labeled as a cyclic C2 symmetry. And when we parse the paper, we realize that the size exclusion chromatography and the chemical cross-linking experiment indicate that this protein actually loses a monomer and does not form dimer in solution. And the DLS study also shown that it exists as monomer, not as a homo-oligomer. And the second example is 1VDE, and the introduction of this paper is explicitly stated that this protein actually exists as monomer in solution in contact of DNA, as shown by 2 previous papers. So this indicates that, as the previous speaker has mentioned, sometimes people just mislabel their the homo-oligomeric state of their protein in the PDB database. And this also points out what is actually important to look into the papers, not just the PDB label. And my ultimate question is that what is physiological? Because when we talk about using protein language models to get the co-evolution information of proteins, of protein homo-oligomers, it's only gonna give you information of things that are physiological. It's only gonna give you contact information, co-evolution information, if the protein is actually homo-oligomer in your cell, and it's actually kind of functionally relevant that it stays as homo-oligomer. So I know that for a lot of people, especially computational people, we take structures in PDB as the ground truth. But is it actually the ground truth? For example, crystallography is actually not physiological because it usually has non-physiological pH, cell concentration, and PEG, and also it's sometimes very easy to mess up whether it's a crystallographic artifact or it's actually a biologically relevant interface. And secondly, at first I saw that in solution experiments are actually physiological, but they also are not, because size experience chromatography doesn't usually have the physiological condition. Cross-linking is a physiological condition, but might just link transient, non-specific contacts. And AMR require EM might have the protein at higher than physiological condition as well. And second thing is that I noticed a lot of some of the, like as you can see here, for this protein, sometimes it has more monomer conformation, sometimes it exists more as a monomer, sometimes it exists as more of a dimer, depending on the concentration. And secondly, this protein can form heterodimers. So this will further complicate the co-evolution data you have, the co-evolution signals you have for this protein. In the end, my conclusion is that protein language models learn co-evolution information of homo-oligomeric contacts, scaling and fine-tuning improves the prediction performance. The missing signals can be explained by a variety of reasons, including conservation, lack of homologous sequences, subfamily-specific intercontacts, crystallographic artifact, physiologically irrelevant homologomerization, competing heteroligomerization. And finally, I have two open questions: is how do we distinguish physiological versus non-physiological homo-oligomers, and how often is oligomerization actually functionally relevant? So I would like to thank my group at MIT and thank the Swiss National Science Foundation to offer me funding to do my postdoc in US. And I'm actively looking for independent PI opportunities, so reach out to me if you're interested. Thank you very much.
Thank you very much. One question here.
That was a very nice talk, and I'm very happy that you have been frustrated about the same things that I have been frustrated about. So I had a question. So when you said you see these missing contacts, right?
Yeah.
So have you looked at the types of interfaces between the chains? And because there is this notion of some interfaces as being primary and essential for homo-oligomerization, and then there are these other contacts that come about once those take place.
Yeah, so we'll do some further analysis, but that's like one interesting observation. Yeah, sometimes I think the contacts, you're more likely to see the contact signals if it's very important for the formation of the homo-oligomer. Yeah.
All right, let's thank Zhidian again. Thank you very much.